All right, awesome. It is noon, so we'll go ahead and get started. Welcome everyone to what is our final MSI Thursday monthly seminar of this semester and this academic year. By now, I think most of you probably know, but I'm Hannah and I manage Harvard's Microbial Sciences Initiative, or MSI. And we've been hosting this monthly seminar series virtually for the past year. And before we introduce Dr. Kathryn Kauffman, with whom I was fortunate to overlap while I was at MIT, I'll first turn it over to Charles to introduce who's co-hosting us today.
Hi, thank you for coming, everyone. My name is Charles, and I'm the president of the MIT Microbiome Club. And the MIT Microbiome Club is the student-run branch of the MIT Center for Microbiome Informatics and Therapeutics. And the purpose of the club is to bring together inquisitive members of the Boston community in the emerging field of microbiome studies and microbiome-based medicine. We typically host a seminar every month, and if you're interested, you can sign up for the mailing list via the link I'll post in the chat. And thank you to the Harvard Microbial Sciences Initiative for graciously allowing us to co-host Dr. Kauffman today. And with that, I'll let Hannah introduce our star guest for today. Thanks.
Awesome. Thanks, Charles. So it just makes so much sense for the various microbial science organizations in the greater Boston area to collaborate. So it's fun to end the year with a co-hosted seminar. As is tradition, when possible, we allow the nominating party for the seminar to introduce them and share their perspective on why they make an exciting seminar for this series. Now, like I said, I already knew that Kathryn would make a great seminar, but I was thrilled to get a specific nomination from Libasha Kelly. So I will turn it over to you, Dr. Kelly, to give that introduction.
Thank you so much. So my name is Libasha Kelly. I am an associate professor of systems and computational biology at Albert Einstein College of Medicine in the Bronx, New York. And I am delighted to introduce Kathryn Kauffman for today's MSI seminar. Kathryn got her BS in biology from Old Dominion University and then moved to MIT's Department of Civil and Environmental Engineering for her doctoral and postdoctoral work in the lab of Martin Polz. In the Polz lab, Kathryn began to develop a truly impressive body of work surrounding her interest in fundamental questions of how phage-bacteria interactions are structured in natural microbial populations. So noting first the lack of genomically resolved model systems that take into account the immense natural diversity of microbial communities, Kathryn developed and executed a 3-month daily time series collecting phages and bacteria from the ocean off the coast of Naha, Massachusetts. And should you want to question her dedication to the cause, I want to point out that she carried out sampling in the middle of a hurricane to keep this time series intact. The value of this model system that she developed is immense and has produced beautiful papers and novel concepts that are going to change, I think, how we think about phage-host interactions in the ocean. Kathryn is also a wonderful colleague and a friend to me, and my own ideas about microbial ecology have been greatly expanded through our many conversations and late nights and probably one too many Cambridge bars. And I am beyond excited to see how Kathryn's going to be developing her ideas in her new lab at the University of Buffalo. And with that, I am really looking forward to hearing about resolving the structure of phage-bacteria interactions in the context of natural diversity. And I turn it over to you, Kathryn.
Woo. Hello, everyone. And thank you, Libasha, for that really lovely and generous introduction. I really appreciate your nominating me for this opportunity here. And I also want to thank the organizers of MSI, Hannah and Julia, for extending the invitation, and also to Charles and the MIT Microbiome Club for this invitation. It's really an honor to be invited and a pleasure to have the chance to share this work. So with that, I'm going to jump in and share my screen. And can everyone see the screen?
That looks great.
Yeah. Great, thank you. Okay, so I am going to talk about the work that I did as a PhD student and postdoc at MIT in the lab of Martin Polz, working in the context of marine systems. And though now I'm in School of Dental Medicine at the University of Buffalo, transitioning into starting a group working on phage-bacteria interactions in the context of the human oral microbiome, the work that we did here in marine systems really informs the approaches and perspective and how we think about what we'll be doing in the context of the oral microbiome. So I think what we found out here is really broadly applicable across systems. So viruses are all around us. They're in the ocean, which I'll talk about today. They're in the sediments and soil and swept up into the atmosphere and raining down on us. They're in the foods that we eat. They're on us and in us, and they're shaping the ecology and evolution of all cellular life. And most of those viruses are thought to infect bacteria. They're called bacteriophages or phages. And here you see a cartoon of a phage on the surface of a gram-negative bacteria. And for this to have happened, that phage had to have been in the right place and right time to find its host and then bind to receptors on the outer surface of the bacteria. It now has to inject its genome to get inside the bacteria, and then it has to evade defense systems of the bacteria in order to go on and replicate. And we can see here what some of the outcomes of that replication might look like here, where we see a burst bacteria in purple and the replicated phages in green. And not all phage bacteria interactions have to go down this way. This is a killing interaction, but of course phages can form long-term associations with their host. But today I'll be focusing on killing interactions. So you can imagine all the different outcomes and impacts that such an interaction might have for a microbial system, including changes in the way nutrients are cycled, changes in the patterns of horizontal gene transfer between microbes, changes in the structure and function of that community, because whoever that was is now no longer part of that community and doing whatever they were doing. So for these reasons, phages offer important tools in, for example, bioengineering and phage therapy, and are of great interest for that, as it's thought that phages can be used to, for example, remove specific members of a community or deliver cargo into specific members of a community to alter the function, or in the case of phage therapy, to remove specific components of a community or system that are pathogens. But these phages are also shaping the composition and function of microbial communities when we're not paying attention and without any interest in our applications, from the ocean to the human microbiome. And to understand the impacts of these phages and to be able to use them as tools, it's important to therefore understand who's interacting with whom, which phages are interacting with each host. That's a key element to both of these broad areas of interest. But unraveling who's interacting with whom remains a challenge. And so that's because these interactions are specific. So here I'm going to highlight, and the members are diverse. So here I'm going to highlight about the bacteria being genomically diverse. I'm showing you here pairs of bacterial genomes, and we're looking at their genome sizes on the Y. These are all in the same species of bacteria, and on each of these pairs shares an identical HSP60 sequence, but you can see their overall genome size can be up to 20% different. And we're learning now that much of this difference in bacterial genome sizes is in mobile elements that have to do with defense against phages. So the bacteria are genomically highly diverse, and so are the viruses. Here is an example from some work by Gregory et al. Looking at the global ocean virome, and only 10% of the viral populations that they identified in that work could be mapped to known groups of viruses. And overall, you can see there in panel D of that figure that the vast majority of those have unknown hosts. So the diversity of both bacteria and viruses is very high. These interactions are specific. So how can we learn about the major patterns? What are the general principles that can guide our understanding of how these interactions are structured in complex microbial communities? And getting at that was the goal of the group at the time I was doing my PhD and postdoc. And so what I'll share with you today is an intro to phages in marine systems, an intro to our model systems, which includes a community model system, the naha collection time series, the hosts that we focused on, which are the vibrio, and their goal there was to capture ecologically representative diversity. Of course, the phages, and there our goal was to capture local viral predators. And then I'll talk about our findings. And starting with this overall picture of the interaction structure in the wild. So the takeaway is that the interaction structure in these systems is sparse and modular. And there's two main patterns that come out of this work, and I'll focus on those next. The first of those has to do with broad host range phages. And a major takeaway is that we've been systematically missing these broad host range phages that are important components of phage-bacteria interaction networks in the wild. The other major signal that came out of that work was about narrow host range phages. And the takeaway there is that they're likely infecting far more broadly than we think they are. So to jump right in, viral infections in the ocean are affecting biogeochemical cycling on global scales. Here you can see on the left, this image with a much expanded photic zone. You can see the light blue is 100 meters and the dark blue is the deep ocean, 3,700 meters. And we're focusing in on these trophic webs in the surface ocean, where bacteria are major drivers of carbon fixation and nutrient cycling in the ocean. And these bacteria have two major groups of predators. The small eukaryotes that consume bacteria and cycle them up into larger and larger size particles that can then be taken up the food web or exported to the deep ocean. And the other major group of predators are viruses. And we saw earlier what can happen when the viruses infect the bacteria, and that is the nutrients are released and contribute to regenerative production and cycling in the surface ocean. If we zoom in on a drop of seawater here, we can see in this epifluorescent stain of viral particles, where a stain has been added to intercalate into the DNA, that there are many little particles here in the background. These are the viruses. And then there are some smaller sized particles in the foreground. Those are the bacteria. Larger sized particles in the foreground, those are bacteria. And so all in all, there are about 10 million virus-like particles per milliliter of seawater. That's about order of magnitude more than bacteria. And again, as I mentioned earlier, most of these viruses in the ocean are thought to infect bacteria. And that would include, for example, the myoviruses, cyphoviruses, potoviruses that we think of commonly when we think of bacterial viruses. But just from looking at this picture, you can appreciate that a lot of questions come to mind. Who's actually there and who's interacting with whom? And to get at that, we use cultivation-based approaches to draw out individual bacteria and viruses from samples like this and ask, what is the structure of the interactions? And to do that, we established the naha collection time series with the vision that it would be a foundation for integrating viral interactions and community dynamics, knowing that initially we would be focusing on only a small subset of information from this whole, but envisioning that once we had hooks into that system, we could then generate hypotheses and have the materials to go back and test how viruses are shaping microbial community dynamics. And what we did here was to collect over 93 consecutive days, daily samples of DNA for sequencing, daily cryopreserved samples of the microbial community for resurrection from our freezers, daily live concentrates of viruses, and other metadata and temperature and nutrients. And one of the first analyses to come out of this work was led by Antonio Martin Platero and Brian Cleary. And they were looking at 16S and 18S amplicon time series of the microbial communities. And what they found were there were cohesive communities of microbes that were changing over on the order of weeks throughout these 93 days. And what I'll focus on now are 3 time points in that time series where we went in with cultivation-based methods to understand which viruses were interacting with which bacteria and what broader general principles we could learn based on that. The hosts that we used to do this were the vibrio bacteria. They're an ideal model for linking phage interactions to host ecology and evolution, and we met them earlier in this figure where we were comparing closely related strains and their differences in genome sizes. And these vibrio are ideal hosts because they're ubiquitous in marine systems. They're large and fast-growing copiotrophs that are important in coastal ocean nutrient cycling. They're ecologically and evolutionarily well characterized, and we can readily isolate hundreds of new co-occurring strains from multiple closely related species in the genus by just going out to the beach with a bucket. And the way that we capture them from subhabitats is to then pass that bucket of seawater through sequentially smaller pore sizes of filters. And here I'm showing you what we might catch on a 63 micron filter, a 5 micron filter, 1 micron filter, a 0.2 micron filter. That lets us access these different subhabitats in the water column. And then from each of these, we isolate and plate onto a selected media that allows the outgrowth of vibrio from the whole community. And with these isolates, we can then sequence marker genes or genomes and make phylogenetic trees. And this approach was developed by Dana Hunt and Lawrence David, who working in Martin Polz's group and in Eric Alm's group. And by putting together this habitat information with the phylogenetic structure information, they were able to discern populations of vibrio, which are analogous to species or representative of species in this local environment. And so by doing this, we can access about 10 or so, 10 to 15 species of vibrio that are out there in the coastal environment. So using this approach, I come now to ask the question from the bacterial perspective about what these virus-host interactions look like. So as a reminder, there's 10 million virus-like particles in a mil of seawater. How many phage predators is each bacterial strain living with? And our approach for getting at this was to expose many bacterial strains from a single sample, isolated the way that I just described, to co-collected viruses to determine the strain level predator loads. And again, this was at these 3 time points in the time series. And so each of these isolates, I grew up exposed to virus concentrates from the same day, plated them in agar overlay, and where there were viruses that could kill one of these hosts, a plaque would form that represents a place where one virus infected one bacterial cell, replicated, produced progeny that could infect neighboring cells, and thus produce a clearing in the agar overlay, which represents effectively a colony of viruses that we could then purify a virus from, just like you can purify bacteria from a bacterial colony. And so for each of these days, I exposed about 400 strains to these virus concentrates. Here you see day 222. This is a phylogenetic tree of the strains from day 222. This is an ordinal day, so that was in July of that year. So, and each of the color bars that are showing here represent the sequenced genome, represent a sequenced genome for that strain, and the color represents the population. And so why did we only pick these select representatives to sequence? Well, it's because those were the ones that were killed by phages. And so on this outer ring, I'm showing you in black were strains where after this exposure, there were no phages detected for the strain. And then if there's a color bar in that outer ring, then that shows that there were phages infecting that strain. And what you can see is the majority of strains did not have any phages infecting them. And where they did, there's usually a green mark, which means there was just one plaque in that sample of seawater that we exposed. So doing this for strains from the other 2 days, representing about 1,400 strains that were exposed to phages, we found that overall, this pattern holds. The number of strains can vary from day to day in terms of how many are infected, but overall, the predator loads you see is mostly green bars, so mostly very low predator loads. So on the order of less than 67 viral predators per liter, which if you remember, there are 10 to the 10th viruses per liter of seawater. So that doesn't seem so bad after all now. So this really raises the question of encounter rates. It suggests that encounter rates would be very low, and raises the question of whether the phages are broad host range then to compensate. Is each of those green bars that we observed on these trees representing a phage isolation, is each one of those the same phage that we've just isolated many times and has a broad host range, or are each of those a different phage? And so to get at that, we now take the other perspective, that is to take the virus perspective and ask about the host range of the viruses. And to do that, I isolated one virus from each of the strains where you see a color bar in the outer ring. So that was about 250 phages. And we asked from the phage perspective, now there's about 1,000 vibrio in a mill of coastal seawater. Are local phages able to infect broadly across all of these genotypes? And our approach there was to assay the phage host ranges on many closely related bacteria to determine the killing profile. So those 250 phages back onto the about 250 bacterial strains that each one of them came from. So that resulted in a large-scale phage host range assay. And what I'm showing you here now is in columns, the phages, in rows, the bacteria, and each color marking represents a kill that we observed. And these are ordered by map modularity. This was a method developed by Flores, Poisson, Valverde, and Weitz to look at phage-bacteria interaction networks. And each of these is colored as belonging to a module, so a group of phages and bacteria that are more interacting based on these host range profiles. Looking overall at this matrix here, I'm just highlighting some of the properties of this matrix. We see that it's overall modular. But the most striking thing about this matrix to us was its similarity to another matrix that was done in the late 1970s, and that was the Moebius-Nottkemper matrix, where you can see, if you just look along the lines, that overall the properties of these matrices are very similar. And in analyzing this data set again in 2012, Flores, Valverde, and Weitz found that it was a modular network, which was different than what they had found in a large-scale meta-analysis of other phage-bacteria interaction networks. So this was a noteworthy observation. But the question kind of remained of what underlies this observed modularity. And unfortunately, at the time that Moebius and Nottkemper did their work, they didn't have access to sequencing like we do now. And so with our matrix, we were able to add genomic information to these analyses to understand what underlies the observed modularity. So that's what we'll look at now. So I'm going to show you the same matrix, but now reordered in light of that information. And so the rows represent bacterial genomes again, and now the colors on those leaves represent the bacterial species. So in yellow, down here we have Vibrio lentis. Here are some Vibrio briogani, for example. Along the top, we have the phages. I have manually organized these into morphotypes. So I've clustered together the myoviruses and clustered together the podoviruses, for example. But within that, if you see the color marking of a little circle, that represents a group that's defined as a genus using a tool called Victor. And so overall, I just want to tell you a little bit about these phages. They're all double-strand DNA phages. We have diverse morphotypes. Their genomes range in size from 10 kb to 350 kb, which were the smallest and largest vibrio phages at the time of this study. And they're genetically diverse, about 170 species of phages identified here. Okay, so now mapping on the information about the kills. Oh, and I want to highlight that this characterizations of these phages was done together with Julie Brown at the time in Liebish's group, and now at Bigelow Labs, doing this characterization of phages in this group, in this collection. So now mapping those interactions onto the matrix, I want to draw your attention to some major patterns that we found. And the first is that there are modules that are defined by viral taxa. That is, groups where the phages were just in a few genera. And so, for example, you can see here in blue, this first set of columns. Those are phages in just a few genera, but yet hosts in many species. We also, however, had modules that were defined by host taxa instead. And so we have phages in multiple genera, but hosts in only a single species. So these are the light green cells here, and looking across here, those are all represented within this section, and those are all infecting Vibrio briogani. So these are diverse genera of phages infecting only within the Vibrio briogani. We also have bloom-like modules phages that are in one species and hosts that are of one HSP60 type, and singleton infections, where that phage only ever killed the same host that we brought it in on originally. And so looking at these together, about 40% of all the phages were represented by these singleton infections or bloom-like modules, whereas 40% of all the infections were represented by this broad host range phage module. And so these are the two stories now that I'm going to focus in on to ask what they can tell us more broadly about phage host interactions in natural communities. So first, the broad host range viruses. And the takeaway, like I said before, is that we've been systematically missing some important viruses. And this was work published back in 2018. In this figure here, this is a representation I like that was a figure generated by Joy Yang, where we see the inverted phylogenetic tree of the bacterial host. And you see here represented in the middle nodes that are the viruses. And in this representation, you can see that these broad host range viruses are drawn to the middle because they're interacting with many different species of bacteria in this collection. So the questions we had here are, who are these viruses? The broad host range was a surprise. So who are they? Why have we missed them in the past? And are they common in the environment? And these are a new family of non-tailed bacterial viruses, the Autoleuciviridae. This is a proposed name that's pending with the International Committee on the Taxonomy of Viruses. And they have an internal lipid membrane, unlike the tailed viruses. The podocyphone myoviruses don't have internal lipid membranes. They have genomes with proteins covalently bound at their termini, which only very few tailed viruses that we know about have. They have short genomes, about 10 kb, when tailed virus genomes are about 50 kb. And they have as their major capsid protein a double jelly roll fold. They have a double jelly roll fold as the fold of their major capsid protein. And I'm showing that here in this cartoon from a ribbon diagram cartoon from PM2, one of the few other representatives of bacterial viruses that have this double jelly roll fold as their major capsid protein. So this was really a surprise to find in bacterial viruses. And I'll show you here why. We have this nice figure from Kunin in 2015 highlighting the two major lineages of double-strand bacterial viruses. Two major lineages of double-strand DNA viruses of all kinds. And these are lineages that are based on the major capsid protein fold. And so on the right, we have the lineage that are represented by the caudovirales in the bacterial viruses, and these have an HK97 fold for their capsid protein. And at the time of our study, there are about 2,000 genomes in RAF-seq that had this fold, and bacterial viruses. And in the eukaryotic viruses in this lineage, we just have the herpes virales. But over here on the left, this other major lineage of double-strand DNA viruses have the double jelly roll fold. And again, at the time of our study, there were just 8 genomes in RAF-seq of bacterial viruses that had this double jelly roll fold, whereas among the eukaryote infecting viruses, there's just this wild diversity of viruses and elements that have a double jelly roll fold, including mitochondrial and cytoplasmic plasmids in plants and fungi, including the poxviridae, including the phycodinaviridae, including virophages that are viruses that infect other viruses. So it was really a surprise why there were so few representatives of double jelly roll bacterial viruses at this time. And what we found in characterizing the Autoleuciviridae was that these are likely often systematically missed because of operational biases. And I'll highlight 5 of those now. So the first has to do with them having lipids. Lipid-containing viruses are chloroform sensitive. And as I mentioned, the tailed viruses don't have lipids. So it's safe to treat samples of tailed viruses with chloroform, generally speaking, as most are not sensitive. But here for these non-tailed viruses, that can be detrimental. And I'm showing you here the evidence for that. So on the left, we have comparisons of tailed viruses that have or have not been exposed to chloroform. And on the right, we have the non-tailed viruses exposed or not exposed to chloroform. And on the height of the bar represents the titer. And so what you can see is on the left that there's no difference between being exposed or not to chloroform if for the tailed viruses. But on the right side, for the non-tailed viruses, being exposed to chloroform significantly drops the infective, the titer of those viruses. And so those samples will be lost where they're treated with chloroform. Viruses having a lipid also have lower buoyant densities, and this matters because in processing of samples for isolation of bacterial viruses, often density gradients are used and specific densities are targeted that are representative of the majority of known tailed viruses. But the non-tailed viruses with lipid will sit higher in these columns. And so when the higher density tailed viruses are targeted, then these low density lipid containing viruses will be lost. A third bias has to do with their genomes having covalently bound proteins attached, and these require protease treatment during some DNA extraction. So I'm showing you that here again, as above on the left and right, comparing tailed viruses and non-tailed viruses with or without protease treatment. And what we see is that for the tailed viruses, there's no difference between being treated with proteases or not in terms of the recovery of their DNA and extractions, versus for the non-tailed viruses, we lose the band if we don't treat those samples with proteases. And this fourth bias has to do with not these biochemical properties, but with a growth phenotype. And that is many of these non-tailed viruses have slow plaquing. So after an agar overlay is performed or is made, it can take some time to see the plaques. And so I'm showing you here on the X is time post-virus exposure, that is the days since we made the agar overlay. And on the Y is the fraction of infections that are observed. And we're comparing the blue are the tailed viruses and the orange are the non-tailed viruses. And what you can see in the blue is that within a couple days of doing the agar overlay, nearly all of the infections that you'll ever observe are there to be seen within a couple days. Versus for the non-tailed viruses, it may be several weeks before a plaque forms. And by then, your plate is maybe in the burn bin or long gone. The fifth bias that is not, I don't have a cartoon for, it has to do with their genome size. So they're 10 kb, and in metagenomic assembly pipelines, for example, if quality cutoffs are set around 10 kb threshold, then these viruses might have made it through this gauntlet all the way through to this point and then might be lost. So they may be there, but never be noticed, even if they've made it this far. So they really are running a gauntlet to get through and be noticed. And so this explains maybe why we've been missing them or why they're underrepresented in many culture collections. And this discovery at this time was really especially fun because there had been recent work by Jennifer Brum, Ryan Schenk, and Matthew Sullivan entitled Global Morphological Analysis of Marine Viruses Shows Minimal Regional Variation and Dominance of Non-tailed Viruses. And they wrote, surprisingly, given that the majority of cultivated bacterial viruses are tailed, non-tailed viruses appear to numerically dominate the upper ocean as they comprise up to 92% of the viral particles observed. So our work suggests that many of these non-tailed surface ocean viruses may be double jelly roll viruses infecting bacteria. We're not saying that they are autoleukiviridae, but we are suggesting that more broadly viruses with these properties will be lost. And in extensive additional analyses that actually I did working with Libesha, we found these in many more groups of bacteria and archaea, and I'm not going to share this here, but if you're interested in this, I hope you'll enjoy that work from that previous publication. Okay, so we think this applies not only in oceans, but more broadly. So in general, this has told us that these broad host range viruses, which we didn't expect, are representative of a group that we're systematically biasing against, and thus also suggests that overall our expectations about phage host ranges may be skewed based on what we've been cultivating. So now to turn to the other major signal from the network, that is the narrow host range viruses. And again, I mentioned the takeaway here is that these viruses are infecting more broadly than we think. Looking at them on this representation here, now we see these are the nodes of virus nodes that are drawn in around the edge of the tree because they're only infecting a small number of strains or one strain. So this raises a question of what are the genomic relationships between all of these viruses if they appear to be so narrow host range, and what do their genomes reveal about how their narrow host range strategy can be successful. And this story is work with my co-first author, William Chang, at the Einstein College of Medicine in Liebish's group. And this manuscript has been submitted and is currently under revision. So zooming in to understand what's happening here with these phages, we'll look at the level of genera. So, and I'll just make a note that these genera are genera as defined using a tool called Victor. So we see that these genera of phages are comprised of members that have singleton type infection profiles. So what I'll just show you here is a couple examples of different genera with this pattern. So each of these rows represents a phage now. So this is the phage name. The column represents the bacterial host, and the color represents the species of bacteria that was killed. And so what you can see generally is most of these phages are not overlapping with each other, generally speaking, in terms of their killing. Here's another example from a genus of myoviruses, where again, we see that there's very little overlap, except in cases where here I've indicated with the bar, and those are cases of viruses in the same species. So generally speaking, when we see high overlap, those are representing viruses that are identified as in the same species. And we see the same pattern here, looking at a large genus of siphoviruses, where again, overlaps are represented as phages in the same species. So our takeaway here is that phages in the same species are largely overlapping and killing with each other, but not generally with phages in other species in the same genus. So to look at this a little bit more in depth, within phage genera, as we were observing, the overlap in killing is low. But now if we put the phage genome diagrams alongside for each of these phage rows, we can have a look now at the amount of recombination that is happening. And so just to orient you to this, each one of these genome diagrams is comprised of block arrows that represent predicted open reading frames. They're colored based on being in shared protein sequence clusters. So all of these open reading frames have been clustered and colored based on being in the same protein sequence cluster. And you can see that overall there is a conservation of gene order and synteny in the structural genes. That's largely these genes on the left are the structural genes that are making them, in this case, I think it was siphoviruses. But if we now take a look at some specific example genes here, we can qualitatively see, for example, this larger green colored block gene that appears in non-nearest neighbors, patchily distributed throughout these genome diagrams. We can see that there is recombination between phages within genera that are not overlapping in host range. And to look at that more quantitatively, we can look only at the aligned conserved regions within these groups and ask about the amount of recombination relative to mutation in terms of driving sequence divergence between these phages. So here what I'm going to show you, or what I'm showing you, is looking at species from the collection. Each of these dots represents a species. Each of these points represents a species in the collection where we had enough members to compare. And what we see is that in quantifying the effective change in nucleotide diversity due to recombination versus mutation, that this is always over 1. And so recombination is an important driver of nucleotide divergence between phages within species. Now, that seems reasonable given that we saw a lot of overlap between phages and their host ranges. So that means one can infer they're getting into the same cell and having the opportunity to recombine. But when we do the same analysis for at the level of genera, we see that there are still many cases where the contribution of recombination relative to mutation is also over 1. And so this suggests that there is also recombination that is happening between phages within genera that are non-overlapping in host range. This is a little bit less intuitive given that we don't see them co-infecting inside the same cell. So it raises the question of how is this recombination happening? So to zoom out a little bit more and ask, well, is this happening? Do we see a pattern like this between phages in different genera? We can look here overall at the amount of co-infection that we observe overall in this matrix. So what you're seeing here is Each row and each column are phages, and every point that's colored in is showing that that pair of phages is sharing hosts. And along the diagonal, it's all filled in because each phage shares a host with itself. You can see some blocks around the diagonal that represents phages that are in the same species, which we already saw have a greater overlap in host range. But then if we now zoom in on a particular case, you see a column here that's a particular potovirus. And we compare now the amount of shared sequence to phages that are in the rows that are siphoviruses. We're going to ask how much sequence similarity is there between those that have no overlap in their host range. And I'll show you this by showing you a circular diagram representation of the phage genome with blast sequence similarity mapped onto that. And so here we see in this backbone, this is this potovirus. And then mapped on here are different siphoviruses that don't overlap in host range with this potovirus. And we can see that there is up to 100% sequence identity over large fractions of the genome of this phage, though they are not overlapping in their killing, they're in different genera, and they're in different morphotypes, and yet they're showing extensive sequence similarity. Now, on the right side here, I'll mention these are the structural, so it makes sense. One is potovirus, the others are siphoviruses, so it makes sense that the genes are different in those regions, and so the gene areas that are shared are those that have to do with replication or interactions with hosts. And this is similar to what's been observed with P22 and its similarity to the lambdoid phages. So this is a pattern that's been observed previously in the literature, but one can appreciate when seeing this, the impact that this might also have in doing recruitment to reference viral genomes and attempting to make inference about which viruses are present in a given sample if we see this extent of overlap or sharing between phages that are otherwise potentially quite different. Okay, so how can we reconcile these observations of narrow host range and low overlap in killing with extensive recombination? That is, how do these phages have the chance to recombine if they're not sharing hosts? And so now what I want to share with you is a simple model that we propose that brings together our observations together with historical literature and emerging information from the current literature that we think provides a helpful perspective for answering this question. And I'll present it in a couple pieces. The first piece is that anti-phage defense systems are prevalent in bacteria. This was synthesized so nicely in a review by Otto Bernheim and Rotem Sorek in this paper called The Pan-Immune System of Bacteria, Antiviral Defense as a Community Resource, where they wrote that even in closely related strains with otherwise similar genomes, the composition of defense systems against phages can drastically vary, as demonstrated in figure 2. And here's their figure 2, where in A we have E. coli and in B we have Pseudomonas aeruginosa. The rows represent different strains within those groups. The columns represent different defense systems. And what you can appreciate is that different strains of E. coli or different strains of Pseudomonas aeruginosa can be very different in terms of their carriage of different defense systems acting against viruses. Working in the Naha collection, in excellent work that was led by Fatima Hussein and Javier Duber, they showed that in between strains in the Naha collection that were the most closely related sets of strains in our collection, the differences in phage sensitivity were due to movement in and out of these types of mobile elements. And so in nature, this was what was driving the differences in phage sensitivity profiles. And this is really nice work has just been out on bioRxiv called Rapid Evolutionary Turnover of Mobile Genetic Elements Drives Microbial Resistance to Viruses. And so the observation here is that intracellular anti-phage defenses are ubiquitous and diverse in bacteria. And the corollary is that phage killing host range must therefore be narrower than the absorptive host range if these systems are working. And in Fatima and Javier's work, they also demonstrated at the bench about this broader absorption of phages relative to their killing, which we infer based on our data, but we didn't test it in this work explicitly. Okay, so this is the first piece. The second piece is that recombinases are prevalent in phages. So 74% of phages in the naha collection encode putative single-strand annealing proteins that are like those used in recombinating and that require between 20 and 50 nucleotide stretch of sequence identity. Previous work has shown that these types of recombinases are prevalent in temperate phages. That was work by Lopez et al. In 2010. So here, I'm just showing a protein network diagram that puts the predicted recombinases from the naha phages, which are shown in white, in relation to recombinases identified by Lopez et al. In different temperate phages. And the colors of their nodes are representative of different families of these recombinases. And so what you can see is that the naha collection phage genomes encode representatives of these different diverse groups of recombinases. So bringing those together causes us to think of reconciling these observations through what we call a cryptic co-infections, or think about it as the cryptic co-infections model, wherein bacterial strains are infected by many different viruses, either through different receptors or also through conserved receptors shared by many different phages. That these bacteria encode defense systems that in some cases will degrade the incoming genomic information from some of these phages. And that therefore, these are, from our perspective, cryptic co-infection events, in the sense that in the laboratory, we wouldn't predict them because we don't recognize that these phages could co-infect because one, we would never see in a killing assay. The ones that are degraded by the phage defense systems, we would not see as having that particular host as a host in a killing assay. So to us, these are cryptic in the context of single pairwise bacteria, single phage interactions. Though in nature, where a bacteria is interacting with many different phages, these co-infection events will be more common. So phages can absorb and enter more bacteria than they kill. Cryptic co-infections are likely far more common in natural microbial communities than we appreciate. And then in the presence of recombinases, the progeny that are produced represent not only the wild types of viruses that we know can kill those hosts, but also the recombinant progeny that are produced through co-infection with viruses that are degraded and can't complete the lytic cycle. So our observations support that these diverse anti-phage defense systems are doing their jobs in the environment and are important in shaping the observed narrow host range killing that we see in this collection. But this work also indicates that phages can quasi-overcome these anti-phage defenses as fragments of their genomes are persisting through recombination with other co-infecting phages. And this is highlighted as one aspect of this beautiful work that just also came out on the archive, led by Damien Peel and the group of Frédéric Leroux at Roscoff on the genetic determinism of phage bacteria coevolution in natural populations, where they show this recombination playing a role in carriage of different counter defense systems in phages in this landscape of bacteria having anti-phage systems and phages having counter defense systems. So this recombination is likely playing an important role in the evolution of phages in this context. This implies also that phages that are in and introduced into microbial communities are recombining far more than we predict based on their killing host ranges. So to close out, some takeaways that I hope you will keep in mind for thinking about phages as predators in microbial communities are that though killing is overall sparse, that is deceptive. The multipartite interactions, or these co-infections, are important to understanding the ecology and evolution of phage bacteria interactions. And though in the laboratory we tend to study them as pairwise interactions, I think there's going to be much to be learned by trying to tackle these multipartite interactions to understand the dynamics. And importantly, from an applied perspective, introduced phages in the context of bioengineering or phage therapy may be recombining with local pools of phages far more than we appreciate if we are basing our estimates of their interactions only on killing assays rather than adsorption assays. So there is likely undetected genome diffusion of phages that are added into systems, into local pools of phage genomes. So it's important in the laboratory to consider both killing and absorptive host ranges. And of course, ecologically distinct and important phages are being systematically missed, and our current assumptions about phage host range are skewed by the dominance of readily isolated tailed viruses in culture collections. So it's important for us to consider biases in isolation and analyses in our studies of phage-bacteria interactions in the wild. So with that, I would like to just close with acknowledgements of the wonderful people with whom I did this work together. I, of course, want to acknowledge Martin Polz, who I had the most excellent fortune of doing my PhD and postdoc training with, and who made it possible to do all of this work. Also, Libesha, who has been just an exceptional mentor and a colleague and collaborator on all aspects of this work, and to whom I'm also so grateful for. And thank you for this nomination again, Libesha, to give this talk. Really appreciate that. I also want to highlight Eric Alm, who was key in the early conceiving of the naha collection time series. And here highlighted also are Julia Brown, and now at Bigelow, and William Chang at Albert Einstein, who were both in Liebescha's group and who were co-first authors on aspects of this work, together with all my other wonderful colleagues in the group who you see highlighted here. And I want to again thank the organizers and the audience, and I welcome any feedback or questions or comments. Thank you.
Thank you so much. That was awesome. It's really, really fun to hear some of these updates from you. So I have a couple of questions queued up here for you from people who submitted during the talk. And then I'll remind everyone who maybe has one in mind that you can submit them either by the Q&A feature, or if you'd like to raise your hand, I can invite you to unmute and ask aloud. So I'll go with one we've got in the Q&A here first. Sue is asking whether in singleton phage recombination, in what genes does recombination tend to occur?
So we, to quantify it, we were looking at the conserved parts of the genome that were structural. And so we see the signal broadly and have not investigated whether there is an enrichment in particular genes for recombination. I think that is certainly an interesting question, whether it's observed more as selected in particular genes.
Awesome. I see, Mary, that you've got your hand raised, so I'm just gonna invite you to ask your question aloud. Let's see. All right, Mary, you're welcome to ask your question aloud when and if you're able, but meanwhile, I'll go ahead and share another one from the Q&A here. David says, you've spoken a lot about anti-phage host defenses. Are these largely CRISPR type systems, or are there other defense systems that play a meaningful role?
Yeah, so there's really been a burgeoning in that field of discovery of diverse systems that are playing a role. So I think we're talking about many different types of defense systems that are at work in these systems. And the bioRxiv papers that I just mentioned dig into some of the mechanisms for particular ones in a hot collection and also in a related vibrio system. So I recommend those together, of course, with the work of Rotem Sorek's group that gives also a broad overview.
Awesome. Okay, we've got a question from Gabrielle.
Hello?
Hi, we can hear you.
Oh, perfect. Really great talk. I'm not sure where I missed this part, but did you consider the population size of the bacterial population in the recombination event, because you can potentially you can have a high recombination rate if one of the specific isolate is in a very dominant population, you know. Or I'm not sure whether you try to connect that part.
We haven't looked at the relative abundances of any of these groups, but we did observe that there were blooms. So I do think it's true that bloom dynamics, where the population sizes increase, are important, and that would increase the number of phages that are preying on them, and then increase the potential for co-infection. So I do think that that is an important aspect of the dynamics. I think that's a great perspective.
Thank you.
Thank you.
Thanks for that question. Hyman Hartman's asking, have you looked into the role of orphans in the interaction between the bacteria and the viruses?
We haven't, although the work that I mentioned by Damien Peel and Frédéric Leroux, they looked at understanding the genetic mechanisms of counter defense in phages for vibrio in a very similar system. And a nice thing about having these many closely related genomes is that facilitates annotation of genes. Once we have a hook on one of them that we can annotate, then we can propagate that through to closely related genomes when we see islands that are variable between closely related phages. So I think though we don't know specifically in the system, I think we have the material to translate that from a related system and understand that better.
Yeah. Suresh asks, did you check recombinations in the presence of any stress conditions? I'm presuming stress on the host.
Yes, we have not done any bench work to evaluate the recombination. All of our observations about recombination are based on genomic signal alone. So the next exciting thing to do would be to test those in vitro, as you're suggesting. I think that's exactly what we would like to do next. And so then asking whether we see shifts in the likelihood of that happening under different conditions is very interesting.
Totally. That's actually really related to a question that I was thinking about, just in terms of the logistics of trying to study these cryptic co-infections, and whether you're starting to think about how one would go about that in the lab.
Yeah, so I think in the work that I mentioned by Fatima and Javier, they look at adsorption. And also in that work by Damien Peel and in Frédéric's group, looking at adsorption of vibriophages broadly at high titers, rather than just looking at killing. So I think we start there to first. It would be nice to have the adsorption matrix. We right now have the killing matrix.
Yeah.
But what we'd like to have on top of this is the adsorption matrix. And I think that would be kind of a first step and focusing that down.
Yeah, definitely. Cool. Someone's anonymously asked, recognizing that and why you focus on vibrio species, have you ever checked these vibriophages for the ability to infect other bacterial species, either lytically or cryptically?
No, we haven't. And we did bring in some vibrio species that, well, okay, let me take one step back. That's not 100% true, because in our selection, we also caught some Shewanella and some Enerovibrio. So there are a few phages that will cross-infect across those groups. So that wasn't intentional, it's because they made it through our selection. But we haven't asked about these systematically by targeting other groups. We haven't done that.
Great. Annie's asking, well, first says great talk, and then asked, are the recombinations recent or ancestral? How to differentiate recombinations from conserved regions, if there are any?
Right, these are the challenges. I think the fact that we see the signal of recombination being more frequent than than single base mutations as the driver is telling us that they're in ecologically more recent time, just that we can still have enough homology to see, or that we have homology to have the recombination. So that suggests they're in recent ecological time.
Cool. Yolanda's asking, do you have an idea of what the frequency of successful co-infection is? How long does an average infection cycle last? And how likely would it be that multiple phages could adsorb and inject their genomes at the same time? I guess the logistical feasibility of these co-infections.
Yeah, those are nice things to think about. I would say during rapid growth, we can think of them as being similar to E. coli phage cycle time periods in terms of replication. So in the order probably 20 minutes. So rapid, but the moment that things become nutrient limited, we might enter states of pseudo-lysogeny or a holding pattern. So doing those tests under states of nutrient limitation, I think would be important and environmentally relevant. And so I think what we can infer about the speed is very much dependent on the conditions we're testing it. And though we haven't explicitly tested it, I would be surprised if we didn't see a range. We noticed with the broad host range non-tailed viruses that they infect and kill late, but I don't know that we've resolved, and Hannah worked on these phages also, and maybe we'll know that it's actually the adsorption that may be delayed in some of these cases rather than a delay in time to kill.
Yeah, I think that's going to be really interesting to eventually see. I think some folks are going to work on that in Vienna, but I think that it's going to be really intriguing as to what it is, why you weren't seeing some of those killings, the lytic killing events show up on the plates until so late, whether it's a physiological change that's allowing for adsorption at that stage, or whether they were just, the genome was hanging out inside and then something triggered that too. Because yeah, related to Yolanda's question, I was thinking about potential interactions with prophages that were already sort of in the genome, and that would allow, if a new infection cycle happened, that you could also potentially get genetic recombination with existing integrated prophages.
Yes, although we did not see much signal for mixing with prophages in the Vibrio
interesting.
genome.
Okay.
So we did look at that, and they do seem to be distinct populations that are not doing much recombining with resident prophages.
Okay, cool. Alex is asking, and then I know we're a couple minutes after the hour here, but I'll pose a few more questions if you're okay to stay. Alex is asking, it says, fascinating talk, thank you. I was wondering if something like meta-high-seq approaches are able to reveal cryptic co-infections that are not observed in the killing assays, or maybe are these events too transient or elusive to be observed like this?
Yes, I think it's very true that those approaches are going to be very important for revealing these happening in the wild. I think the challenge is that in those cases we're depending on it to have happened rather than systematically testing it. So, but maybe applying those similar methods to laboratory systems to ask whether we see it might also be of value. But I think it's true that information is likely out there in some data sets already to some extent that would be worth exploring.
Yeah. Yeah, Ryan's asking a question also that's a little bit related to a previous question about the range of the host range of these phages, but asking about whether you've thought about the influence of non-vibrio host phage interactions outside the matrix. Could these be superinfection events that lead to recombination as well?
Of whether there are other hosts, is that?
That's what it sounds like whether interactions, other host phage interactions outside of what's being captured in the matrix, I suppose on either the phage or the host end would be influencing and leading to recombination also.
Yes, I think that is an important consideration. And so in thinking about our model more, and I didn't talk about this here in the talk, but there was work done by John Waterbury and WHOI that showed that the strains of bacteria, in that case it was cyanobacteria, that were the most sensitive to phages are also the rarest and the fastest growing. So there seemed to be a trade-off. So what that implies is that in our collection, if there are very sensitive hosts, we might not have captured them in our collection because they would be more rare than vibrio that are resistant to phages. And so our collections, culture collections in general, may be dominated by strains that are more resistant. And so it may be true that some of the rare guys out there that are fast growing are more sensitive, and when they bloom, when they grow quickly in response to some nutrient pulse, they become really abundant and become a place where there can be a mixing bowl of co-infections and recombination. So I do think that's an important point.
Totally. That's a really interesting, I hadn't thought about that, and I'll have to read that work though. That's a really interesting point. Well, now that we're at 105, I would love to just ask you for a kind of an overview perspective on, to wrap up. I'm curious whether you think these similar patterns are going to hold in the oral microbiome, or whether they're going to be sort of environmentally specific rules governing these phage-host interactions.
Yes, that's nice. That's nice. So in the oral microbiome, I do predict that there will be a real importance of multipartite interactions. I do think that's going to hold. I think that having co-infections between phages is going to be important across systems. I also think, though, that with human microbiome systems being more dominated by prophages, as far as we know now, I think there will be different aspects to those dynamics that are not well represented in these systems that are more virulent or bloom responsive. So cool. I'll hopefully have a tighter answer to that, and not too far from now.
Yeah,
we'll get to work on that.
This has been really awesome. I think not only did I enjoy it, but the engagement from everybody here and the awesome questions that we got makes it clear that everybody else really did too. So thank you so much.
Thank you to everyone.
It's just been a really strong finish to this year, and it's been really great to have you.
Thanks a million. I really appreciate it a lot. Thanks for the opportunity. Thanks for the feedback. Thank you for the nomination, Levisha. Thank you for the co-hosting and the hosting. I feel very lucky. So thank you to everyone.
Definitely. All right. Thanks to all who attended, and all right, looking forward to talking to you all soon.